Hi, I'm Peter Reinhardt, and welcome to the Johnson & Wales International Symposium on Bread. I'm coming to you from the Hance Auditorium in Charlotte, North Carolina, where we've held the last 4 bread symposiums, and as you can see, there's nobody here but me. And this is where we usually have it. The seats are all empty because this year, thanks to you, the symposium will be presented online, virtually, in our new presentation hall, which is where I will join you in just a minute. Thank you, and thanks for being part of our new virtual Johnson & Wales International Symposium on Bread, presented by Puratos. Welcome again. Throughout the entire symposium, I'll be thanking our generous sponsors over and over again, and ask that you do as well by visiting their booths and pavilions in the exhibitor hall. There you will see lots of bonus content, and you can also make appointments to meet with the folks from these companies that serve our baking community so well. Our presenting sponsor is Puratos, who has partnered with us from the very beginning for all of our symposiums, and it is their support that helped us get this one-of-a-kind gathering of thought leaders off the ground. Please also visit our fabulous flour and milling sponsors, Ardent Mills, Lindley Mills, and Central Milling. Thank you also to our equipment sponsors, the WP Bakery Group and Allied Bakery and Food Service Equipment. And thank you also to our specialty food product companies, Probiotine, Fire Within, Big Green Egg, and Mock Mill. Please check out all of their booths to learn about their wonderful and unique products. And also thanks to our media sponsors, Cook's Country, The Local Palate, the James Beard Foundation, and the Bread Bakers Guild of America. You'll be hearing more about all of them throughout the entire series of presentations. So again, thank you to all our sponsors. At the end of today's presentation, you will also see our credit scroll thanking all of the people behind the scenes who made this event possible, including our production and technical partner, Ganoid Communications, our creative team at Gumbo Marketing, and the many folks at Johnson & Wales University, our hosts for this, our 4th annual gathering. So stick around, if you will. But now it's time to get things rolling with today's presentation, so let's go live. And once again, welcome to the Johnson & Wales International Symposium on Bread, presented by Puratos. Yes, we are live again. Hard to believe. We're wrapping up July. Can't believe it. Time is flying by way too quick. But we're going to have a great Monday. And of course, as I mentioned last week, for those who weren't here, we will not be back this Wednesday. We're taking this Wednesday off, which is usually our demo Wednesday. But next week, next Monday, we're going to be joined by Daniel Leader, the founder of Bread Alone, the legendary bakery in upstate New York, and the author of many award-winning books. And he's going to be talking about the whole journey of craft to scale, and also his efforts to turn his bakery, Bread Alone, into what we call a climate neutral bakery. And he'll explain all of what that means next week, but that's an important part of sort of his vision for the future of his bakery. And then next Wednesday, we will have Carl Worley, the founder of Biscuit Love in Nashville, Tennessee, is going to do a demo for us, showing us how he makes his unique angel biscuits. And of course, everybody who's watching probably has their own biscuit recipes, and there's many ways to do biscuits, and there's many ways to do angel biscuits. So Carl's been getting a lot of notice for 3 or 4 of the different types of biscuits he features. He's going to show us the angel biscuits next week, and we'll get to pick his brain a little bit about the whole world of biscuits. And before I introduce Erin McKenney, our featured speaker today, who I'm very excited to see again, Erin is essentially the record holder now, the symposium record holder for most presentations, which tells you how popular she is and how good she is at what she does. and how much we love having her here. And she's going to be doing a presentation in a minute. I'll introduce her, but before we do, we're going to spin the wheel because we still have a knife to give away, prize to give away for, and this is be our July spin. So Gurmit, do you want to go ahead and put that Wheel of Fortune up there? And we're going to give it a spin. I'm going to put my glasses on so we can see who it is who wins. And I don't know how he does it, but he makes the thing spin around. And here we go. You let me know when it sort of stops randomly. That it? William Romeo, Bill Romeo. Bill is the winner of the knife this week. And I know Bill. And I know he's not, I think he's actually on vacation this week. So I'm going to have to shoot him an email later and let him know that he was the winner. He'll be thrilled. He is a serious home baker. And I'm excited to know that he's going to win one of these knives. And just so you know, I had nothing to do with the spin. It's totally out of my control. Nobody knows all the people on there except I know a few of them. But the thing just sort of does its own thing, and we'll do this again. I've got a couple more knives to give away, so we'll do one in August, maybe do another one in September. So keep signing into those guest books so that you stay eligible, because we'd like to find a home for all of these wonderful knives. That said, time to introduce Erin McKenney, Dr. Erin McKenney from North Carolina State and beyond. I'm going to let her kind of fill in on all the things that have happened since the last time she was with us. Hi, Erin. And I know today, the title of your talk is Disruption Through Democratization. And I'm going to let you explain what that means. I'm going to turn it all over to you and just take it away. For those of you who are watching, if you have questions along the way, Erin said, please write them down, do them in the Q&A. It's easier for me to track them there. But if you want to do something in chat, you can certainly use the chat as well. And I will feed them to Erin either during her talk or at the end. We'll leave some time for Q&A, but we'll work some questions in as appropriate. So Erin, the floor is yours. Go for it.
Thank you so much for that introduction, Peter, and for having me back for this year's Bread Symposium. It's an absolute honor to be included in this incredible community of baking experts, and I've really enjoyed the sessions so far. I find it ironically fitting that we've had to go virtual to present this year's theme of disruption. As Peter said, I'll be talking with you today about disruption through democratization, how citizen science transforms sourdough research. So we'll kick off with, what is citizen science exactly? Citizen science has gained kind of a buzzword status in scientific communities and academia and beyond. At its core, citizen science is a partnership between researchers and the general public, where people who might not necessarily identify as scientists, who can still contribute to scientific knowledge by helping to gather and analyze data. Now, researchers benefit from these partnerships because citizen scientists cast a wider net, vastly increasing the scale of data and discovery that's possible, right? Think just for a moment in a completely baking context, how much time it would take to grow and refresh 40 starters by yourself, 40 sourdough starters. Now scale that up to 100 or to 500 sourdough starters, right? One human, if we did that, that would be all that you did for the rest of your life, right? But with the power of citizen science, by partnering with the public, we can cast this wider net. So we have the potential to, make greater discoveries, to find things that previously were unknown, simply through the power of scale. But it's equally important to me to root any citizen science partnership in reciprocity. I'm very committed personally to translating our research into protocols that anyone can follow at home, and to share our results back to all of our participants. So a few weeks ago, Michael Gansla described bread production at scale, referring to industrial, automated, and high throughput bread production. But I use citizen science to increase sourdough production at a global scale. So instead of maybe vertically, then maybe, across horizontally across the entire globe, by empowering the general public, including students from kindergarten through university, and the general public being museum goers, or just anyone who has ever gone online and thought about sourdough or bread, to grow and study sourdough starters that they can then use to bake bread at home. I've worked with Rob Dunn, Ann Madden, Lauren Nichols, and Leah Shell on 3 different sourdough citizen science projects so far, which have all been pretty wildly successful, not only in terms of recruitment. You can see our global citizen science project, we had 1,000 participants, and then a whole 500 of them sent us their sourdough starters from across the world. And then we launched the Wild Sourdough Project. I've got the coronavirus icon there because we did launch it in April at the very, towards the beginning of lockdown over a year ago, to engage folks who suddenly were at home a lot more than they were used to, or maybe felt like they had more time on their hands than they were used to. Why not learn how to make a sourdough starter? Right? So not only in terms of recruitment, getting folks to participate in our projects, but also helping to build community and conversation among scientists and bakers from the novice to the expert level. And we would consider that kind of community and conversation to be evidenced by the Facebook group membership. 826 of those original 1,000 participants for the global project, and over 2,500 members now who have heard about or participated in or are keen to participate in our Wild Sourdough Project, right? So I'll start with the Global Sourdough Project. Both Michael Gonsla and Rob Dunn mentioned this paper in their talks, so I'll focus on some of the points that they didn't already recap. But briefly, you can see this incredible team of scientists. We did build a veritable complementary pyramid, both of skill sets and of human perspectives, just a fantastic group of researchers to be a part of. On the greater backdrop of our global data set, you'll see there was still a bit of a Western world bias, right? There is still sampling to be done in many parts of the world, but 500 starters had us pretty happy and had the post office pretty busy, shipping all these wild inflated packages from all corner, all places across the world to our labs so that we could study these sourdough starters. So first, when we compared bacterial and yeast communities across all starters, we identified some pretty clear patterns of membership, right? Meaning you can see these patterns of banding in this heat map presented to you. So if you consider, each row, each very thin row of samples is, or of lines across the graph is a single starter, right? So along the left side, you see kind of that family tree of sourdough starters. That's grouping each sourdough starter next to its nearest neighbors. So other starters with very similar microbial membership. And along the bottom of the graph, you can see that we've got acetic acid bacteria, lactic acid bacteria, and yeasts, all different microbes identified in each of these. So we see these clear banding patterns of membership. But when we ran statistical analyses to determine which sourdough feeding practices or other baking practices of sourdough management might explain these patterns, we found that all of those factors that we as bakers consider to be super important actually together only explained about 10% of the data. So a humbling moment as the microbes reminded us that, this is everything that we think about, right? Everything from where geographically your starter lives, to how often and what type of flour you feed your starter, to your age, your sex, how large your household is, whether you have pets, all the things that we as humans might think would be very important. They really only explain 10% of the variation. Instead, Liz Landis found that the interactions between different microbes play a much greater role in shaping communities. So for example, which taxa can outcompete the others for maltose, right? Or for other nutrients in either the flour or, in the developing sourdough starter. So here is a very simplified kind of snapshot of some of those interactions. If you consider Kazakhstania humilis and Lactobacillus plantarum, clearly this was made before the great Lactobacillus renaming scheme. But our red shaded boxes are positive correlations, so microbes that get along well together, and the blue are negative correlations where you tend to see only one or the other microbes. So we saw some pretty strong correlation patterns that indicate either positive or negative relationships that might drive these overall communities, no matter where the starter is kept or how it's kept across the world. And these interactions not only resulted in different community structures that were consistent across geography and across sourdough management variables, but we found that different communities produce specific aromatic profiles. And that to me is one of the most exciting and potentially disruptive findings. One, because it is, again, humbling to realize that in the end, the microbes make a much greater difference than we give ourselves credit for as far as sourdough profiles go. But two, because this link between community structure and aromas may actually affect our aesthetic experience of the bread product, right? And three, because acetic acid bacteria has not historically received a lot of attention in sourdoughs, yet they seem to be major drivers of the aromatic profiles that we analyzed. And I love a good underdog story. Let's give some credit where credit's due. So the same way that your starter multiplies for every answer or every question that we were able to answer for the global sourdough project, we seemed to develop 7 new questions that we wanted to explore. So we have over time developed a number of spin-off projects inspired by feedback that we've gotten from our participants on the original global project results. So for example, when I first started working on the Global Sourdough Project, I also taught natural science at the North Carolina Governor's School in Winston-Salem. I asked my high school students to bring flour from home, whatever flour was most commonly used in their homes or their personal favorite type of flour, so that we could culture the bacteria and the yeasts living in each flour. And we found striking differences between all-purpose versus different whole grain flours. Some pretty clear evidence from homes across North Carolina that regardless of who is distributing or milling your flour, even at the commercial or the generic brand or the name brand scale, the flour that you feed your starter is going to introduce not only different nutrients to your starter, but also different microbes that actually kickstart that community to begin with. I also worked with Ann Madden and Leah Shell to launch Sourdough for Science, a lesson plan that enables students to grow their own sourdough starters from any type of flour they want and to measure the changes. So they're smelling, they're measuring the height and the pH over time for 2 full weeks until those starters can be used for baking. And in the slide, I've hyperlinked our webpage for Sourdough for Science. Any of you who are also interested in education, we went ahead, I partnered with local middle school teachers in the area 2 summers ago now, time flies, 2 summers ago to also align our activities and supporting activities to complement national teaching standards. So again, making sure that sourdough is applicable not only in the home, but also in our schools. Throughout these different efforts, I worked with local bakers like Josh Bellamy at Bolted Bread here in Raleigh. Josh actually baked breads from all of the best 6 starters from each of 3 different middle schools. He baked into, probably hundreds of loaves so that we could show, yes, indeed, at 2 weeks old, your sourdough starter, spontaneously fermented though it may be, it can indeed produce a viable loaf of bread. And we actually had taste tests to see, you have smelled the differences in your starters. Here is how some of those aromatic profiles manifest in the finished product, the loaf of bread that you bake with those starters. And I've also worked with some not so local bakers, including many of you who I've met at previous bread symposia. And in every conversation that I've had with a baker, or multiple bakers at once, I've always been impressed by how many different techniques there are and by how many questions bakers have for me about what's going on in bread. So with all those questions in mind, I partnered with Martha Calvert, who is now getting her PhD at Virginia Tech, to review the literature and to summarize the research on sourdough. Our paper was published earlier this year and is also hyperlinked at the bottom of the slide. And the most prevalent theme throughout Martha's paper, Martha is also a baker. She kept coming back to me as she was doing all the research and just saying, Erin, the research methods just don't reflect baking practices that I see and that I'm using in artisanal baking settings or by home bakers. It just doesn't seem relevant. And that made us wonder, whether the research findings are in fact relevant directly to the general baking public. So to bridge that gap, Martha suggested that researchers adopt methods that do translate readily to the home and that reflect actual current baking practices at all different scales, not necessarily just the methods that are used in research labs, not necessarily the methods that are used most commonly at an industrial or commercial scale, and to study the impacts of common home baking practices on the microbial ecology of sourdough. So in my mind, all of this equates to citizen science. So I fine-tuned the Sourdough for Science protocol to measure flour using tablespoons instead of a scale, just thinking through, I know a lot of us use weight instead of volume, but if I go into the houses, a random sampling of houses in Raleigh or across the United States, more people will have tablespoons than scales. And next, I partnered with 4 local middle school teachers and an undergraduate researcher to compare bacterial succession over the first 2 weeks of a sourdough starter's life in sourdoughs that were grown from 10 different types of flour. So we grew 4 starters each from 10 different types of flour, so we had 40 starters. All this to help address some of the most common questions that I regularly get from bakers. Is it true that you can use a starter as early as 10 days? Do I have to really wait for 10 to 14 days? What difference do these different flour types make to a starter? And as the starters grew, we measured changes in acidity using pH paper that's available and affordable online. And we also smelled the starters every day to describe their aromas. Now, for the global sourdough project, we identified aromas and aromatic compounds using gas chromatography and partnering with a professionally trained sensory panel, who I like to call sourdough sommeliers. But that level of expertise or equipment doesn't lend itself well to citizen science. So this time around, we partnered with Michael Bohm-Frost at the University of Copenhagen to create a sourdough wheel that would help folks to describe what they smell using specific and standardized language. So Michael's lab grew over 100 sourdough starters from all different flour types. It smelled everything, drawing on their professional training, and developed this sourdough wheel after the style of the wine wheel to provide a standardized language. And here you can see a picture of Kristen Kemp, one of our local middle school teachers, using that wheel in action. So across flour types, we identified distinct functional stages of succession that are characterized not only by the sourdough starter's acidity and rise, but also by the specific bacteria present in the starter at that time point. Early on, we saw a lot of plant and bodily affiliates, and the starters smelled mostly like flour. You'll also notice that the sourdough falls when the pH drops because the yeast in the flour aren't really acid tolerant. This is hopefully reassuring news for everyone who has ever grown their own starter and at day 2 or 3 says, I think it's dead. It's not dead, it's just transitioning. Trust the microbes. By day 6, the pH has stabilized at a much lower point, and that low pH selects for acid-tolerant microbes, including yeasts. So the sourdough starts rising again. It's rebounding, and it smells more sour and a bit like fruit, possibly due to esters from those recovering transitioned yeast communities. But then by day 10 to 14, the sourdough community has stabilized. We're ready to bake with it. And we've settled into those, typical acid-producing, acid-loving bacteria that we tend to associate with sourdough. Lactobacillus, but also Pediococcus, also Gluconobacter, which is one of those acetic acid bacteria that I've been rooting for. But even though we saw similar patterns of succession across all 10 types of flour, we also saw each of those distinct bacterial communities and aromatic profiles that were associated with each specific flour type. So as the communities grew toward their more stable, mature climax communities at day 10 to 14, here I have a heat map showing you the relative prevalence of these kind of key players, the bacteria making up the different communities in the sourdough. On the next slide, I have the data presented in a different way, right? So on this plot, each point is a different sourdough starter. The closer the points are, they're color-coded by flour type. The closer the points are, the more similar their microbial communities, right? And what you can see is, one, that each flour type forms its own cluster of a unique community type. There is some difference in overlap and variation across the different flour types. But we also plotted some of the most prevalent aromas and the most prevalent microbes that distinguish those mature sourdough communities at day 14. And so you can see that relationship between flour type, microbes or the bacteria making up the communities, and the aromas those communities produce. This means that even without DNA sequencing, to know exactly which specific types of bacteria live in a particular starter, bakers can potentially select for particular microbes and traits by feeding their starters different types of flour. So, circling back, how does citizen science transform sourdough research? Similar to the Global Sourdough Project, we were able to identify new bacteria and different relationships than were previously known or appreciated in sourdough starters. This offers us, some potentially novel pathways to manipulate the functionality, whether that's flavor or rise or the pH and resulting, sour taste of the breads. And this, I think, is the great reciprocity of citizen science, that by partnering with the general public, we're able to discover more by operating at a greater scale and across more different contexts than we ever could have done alone. And our findings have greater relevance because our protocols reflect current practices. So we're not only transforming microbial ecology through a greater understanding of sourdough, we're also transforming our relationship with the people who make this research possible. So looking ahead, Martha and I are interested in working with Matthew Booker to develop a census-style survey so that we can map the diversity of bakeries and baking practices across the United States and maybe eventually across the world. We hope that what we're fondly referring to even in Dreamland as the Great Baking Census will help us to document and appreciate the wide range of practices that people are using every day to make bread. So stay tuned. We hope to be in touch with a link to that survey sometime in the near future. And that's it, my whirlwind tour of everything that we're learning together about the science of sourdough. Thank you so much for tuning in. At this point, I will breathe for a minute while I check the Q&A and see if y'all have any questions.
This is great. Yeah, folks, go ahead and start writing your questions. I'm not seeing any up there yet. and typically sometimes they'll start to pour in as you have a chance to type your questions. I love that starter shot you have there at the end of your, in that last slide. That guy looks like it's taken off for sure. While we're waiting for other questions to come in, let me ask, I've got a couple, just to get the ball rolling. Oh yeah, the questions are starting to come in, so we'll get to those. But when you give the instructions to your citizen scientists, do you give them a hydration sort of formula, like what percentage of water to flour they should use, and are they all following the exact same protocol, or are they free to kind of go with firm versus spongy starters and stuff?
Yeah, so we, in the interest of standardizing the methods so that we can focus on flour type, we asked everybody to use, equal volumes. So 2 tablespoons of water and 2 tablespoons of flour to start with. And then after, backslopping or removing about half of it, so discarding a tablespoon the next day, then adding in another tablespoon of flour and a tablespoon of water. That just helps keep everybody on a level playing field, so that we can compare their results if we're asking, how does flour type specifically change the bacterial communities and the aromas and the pH and the rise over time. Then we want all the starters to be the same age, and we want to know that we are using about I guess that's 50-ish, right? No, 100-ish% hydration if we're using about equal water and flour.
Well, actually, if you're using it in volume, the water is higher than 100%, because flour is like only about half the weight of water. So
Okay.
it's probably more like about 150% or so hydration. And I'm only asking just in terms of understanding the consistency, and if different starters yielded different results because of hydration. But
Yes.
that's controlled by the protocol.
So we try for that with equal volume. And again, the tablespoon is something that's, just going to be a more commonly available tool that's already in folks' kitchens.
Mhm.
But we do find that across different flour types, and I'm sure people are thinking like a gluten-free flour is going to act a lot different than a glutinous flour, or, a rye is going to behave a lot differently with the same amount of water compared to a whole wheat flour. And we do see differences in those textures and consistencies of the starters. We're just trying to hold all of the inputs as standard as we can, so that, we can, given the available equipment that folks already have in their homes, that
Right.
we can then engage them to ask, questions specifically about age or flour type. But we have, in the Wild Sourdough Project, we've challenged folks to then say, go wild. If you have a question about how does the hydration affect it, then use the same flour type and the same water source, and control all of your other measures, and then just vary the amount of water you put in.
Yeah, so they can run their own experiments, not just the one that you're giving them.
Exactly. So hopefully by participating in the scientific process, we're further empowering people to feel like they can take control, even when we don't know what the answer will be.
I want to come back to that one a little bit later, but before we do, I want to get some of these other questions out to you, because I think that's unique. It's exciting to know that you're actually not only incubating, an experiment, but also incubating future scientists. And I love that, especially with the middle schoolers and things like that. So we'll come back to that one in a bit. But I'll take them in order here. Jean asked, I'm really intrigued by the sourdough wheel. And so I'm going to ask you, will we be able to, can you provide this PowerPoint that you showed us today? Can you make that available to us to put in our archive folder for people that want to be able to go through it again? Because that way we can see this wheel.
Absolutely. And that wheel is also available. If you Google Wild Sourdough Project, then you should be able to find our webpage, and that has the wheel available as part of our protocols that are publicly available.
Really neat wheel. I love wheels like that, that break it all down into these very, sort of subtle differences. Michael Calanti talked about that as well in his presentation. And a couple of years ago, he made the presentation, I think at our very first symposium, where he created his own chart. It wasn't quite a wheel. It was more it was squarish rather than roundish, but it was the same idea, isolating for the purpose of, as he said, to articulate a new language for being able to zero in on these differences. And so, but this really, how many different items are listed around this? It looks like
I haven't counted them recently. I'm coming up with, I think there are 13 super categories in the inner circle.
Yeah.
So, don't be intimidated by all of the ones around the perimeter. You can start with, does this smell bodily or does it smell chemical, right? And narrow it down to your super category. And then within chemical, does it smell like, maybe I'm choosing the wrong one. We can try for a more aesthetically pleasing, right?
Well, I have had people write to me saying that their starter smelled like vomit. So I understand that, but I think that most of the time, if they stick with their starter, they'll pass through that phase and get to something that ends up being much more pleasant at some point.
Yes, absolutely. It was really interesting to see. We tended to identify many more bodily smells earlier in the lives of the starters. So in that initial stage, when you have a lot of grain and body affiliated bacteria.
Yeah, the sweaty feet one is probably the most common one that you hear, about. So that's interesting, because I'm glad to know that my empirical tests have kind of matched up with some of the ones that you've come up with as well. So folks, the chart is there, and the wheel, and I think it's a cool thing to have. So you may there could be a little cottage industry in selling posters of this wheel. They did it with the grapes and they did it with the cheeses. So maybe get that published. Mike Avery says and asked, Dr. Gonzalez said, in sourdough there is no terroir. But you talk about the differences between the starters resulting breads. Can you explain why you have a different viewpoint than Michael?
Oh, so there's no gauntlet thrown. We don't actually have different viewpoints. From what I understand, Dr. Gonzalez was saying there's no terroir in that there's no geographic or regional difference among sourdoughs. And that's exactly what we found in the global project.
Interesting.
That, sorry, San Francisco, with the right, very specific management steps, we probably can make a comparable sourdough anywhere in the world. We just have to figure out that secret special combination of hydration and flour type and temperature and fermentation time.
But you did say that the flour itself, different flours will bring in their own families of organisms.
Yes, and that's what we found. Even though we saw across flour types similar patterns of succession, we did see by day 14, you have clusters of communities that are more similar within the flour type that they were fed. And those similarities in microbial membership are also associated or correlate to differences in aromas that were produced. So amaranth is the most dramatic example. We were calling it hamaranth because it smelled like ham.
Amaranth.
Yeah, we were smelling that amaranth sourdough starter, and we're just like, that's definitely meat. That's ham. I mean, incredible. How do you get such a strong ham smell out of a grain? So that's the linkage that I was speaking to. And that to me is really exciting, because that's a relationship that we can leverage, right? Even without DNA sequencing, we now know that, these across these different flour types that were grown otherwise in similar conditions, those starters, a flour type selects for specific distinct bacteria that produce specific distinct aromas.
Wow. Well, along the same line, Emily's asking, did you say that when you tested flours across North Carolina, the type of flour mattered, but the brand did not? Or did brand also matter?
Great question. There were some variations between the different brands. So again, these are high school students, and we're in North Carolina. So there was a lot of all-purpose flour, just your basic white flour. And it came from the Food Lion grocery generic brand, Pillsbury, what is it? The Pillsbury, gold medal, Lily White, right? But we saw that all the white flours, it looked like somebody sneezed on the plate. I mean, they were covered with lactobacillus bacteria. Whereas we didn't see very much at all of that lactobacillus bacteria in the whole grain flours. There was some variation, but nowhere near what we saw in the white flours. And the all-purpose flour didn't have any yeast growing in it at all, no matter where it came from.
Really?
Whereas the whole grain flours tended to have different yeast colonies growing on them and in different amounts.
Well, that would make sense, I guess, because the yeast would be more on the bran, on the surface of the berries.
Exactly. So it was nice to be able to kind of lay out those mental breadcrumbs for the high school students to be able to put that together, thinking through, how do we process our food? What are we gaining or losing when we use these different processing techniques?
Well, Mike Nolan asks, he puts up a link to something from Tom's Hardware. It's a gadget. Would it help people trying to make sourdough? Do you recognize that in the Q&A from Mike Nolan?
Yeah, I got a screenshot about it. There were none available, and sadly, I was not offered one for free to test and use. But I heard that they were in pretty high demand. It looked pretty sweet.
What is it?
So the Raspberry Pi, it's a small programmable computer. I think they cost $50, and then you can program them yourself. And somebody rigged up a Raspberry Pi based sourdough monitor. So if you have your starter in a container and you have it kind of sealed, you can put probes into it that then collect data automatically and continuously.
Wow. So that would really make you a citizen scientist with a capital S.
Yes, yes, exactly.
More democratization going on here, Erin. That's pretty cool. But for $50, that weeds out the rubberneckers. It's for serious, citizen scientists. But it sounds like a great tool.
Yeah.
I never knew about it. So Mike, thanks for bringing that up. And of course, Mike Avery asked about this wheel. Well, we talked about that. You can get that in a couple of different ways, but we'll have it, in our archives once we get the PowerPoint copied in there. Heike asked, any ideas on how the bacteria composition change once the starter has stabilized? After a month or a year, does changing the flour at this point make a difference?
That's a great question. And it's one on the long list of questions that we're collecting, right? We can't get ahead in this game. Every time we answer or every time I present, I get, 17 million new amazing questions that I want to follow up on. That is on the list of things to look at. Once you've reached a stable point, how much disruption can the starter handle before it shifts completely? How resilient is a starter? And it's something that we don't know the answer to yet. Anecdotally, we hear a lot from folks who have gotten their starter and it has changed over time, or from folks who have shared their starters with other people, right? The donor to the recipient. And then, the recipient uses a different flour type and says, well, it's not the same as when I got it. And I think, well, sure, you put it on a completely different diet. So anecdotally and let's see, kind of speculatively, it makes complete sense to me that, with a drastic change in one or more parameters, that you could alter your microbial community and its behaviors. We haven't put strict quantitative numbers on it. We haven't done a study yet.
I thought about that question years and years ago when Sourdoughs International was selling their little starter packets from all over the world. And you could make 3 or 4 starters and have them side by side and see the difference at the beginning. But after a couple months of, refreshing them and using them, they started to tend to kind of become similar to each other. And so I figured there had to be, even though they claim that they wouldn't change, but I can't imagine how you can guarantee that, because bacteria is going to, the dominant guys are going to win in the end. And of course, I was doing my experiments in San Francisco where there was just so much, wild yeast floating around that it was easy for it to kind of all become what it was. But so that sounds like something for more research down the road. Did you notice? Oh,
I think there's potential, there's the brewery supply stores where you say, I want to make a clone of, this Belgian milk stout or whatever. And then you get a recipe and you use specific malts, specific hops, specific yeasts, right? They've narrowed it down to a specific strain that does exactly what they want. And they know the exact parameters nutritionally and, a temperature and length of fermentation. Mm-hmm. I think with enough of these studies and enough rabbit trails, right, we have the potential for a baking supply store that we may not have regional terroir, but that means that we have greater potential to grow and produce whatever bread we want, given the knowledge and given the adequate and appropriate flour inputs and fermentation parameters.
So that's a great entrepreneurial opportunity for somebody out there who's young and hungry enough to make it happen. Peyton asks, where and when might some of this research be available to us? And are you looking for other samples?
Great question, Peyton. So I have included hyperlinks to in the talk, which again will be made available shortly, to Liz. It's Landis et al., the global sourdough study that I started with, and then to the sourdough. What did we call it? The Sourdough Microbial Ecology Literature Review that was published in the Peer J journal. And that's by Martha Calvert. I'm a co-author on both of those, but the first authors will get you where you need to go.
So by the way, folks, this is great that you're doing closed captions underneath, so you guys can catch some of these names. I don't know if they're all spelled, if the closed caption guys are spelling everything right, but at least you can get a quick reference there. And then, but that first one that you said, there was one, was it Lambert et al. Or somebody?
Landis, Landis
et al. So Landis and others. So if you could just start to do a search set for Landis what was it called? Global Sourdough Study. Is that what it was called?
Let's see. Here, I'll type my answer into the Q&A here. Landis et al. Sourdough e-life. If you Google that and Calvert sourdough Peer J.
So look under, it should be under.
So in the answered Q&A, I've given you Google search terms that will get you where you need to go.
Okay, great. And it looks like more questions coming in along the same lines. Not clear on the 4 sample for sequencing process.
So I think you're referring to this graphic. So let's see, this is a graphical protocol. I think visually, and anytime that I'm working with any researcher at, any experience level, and for myself, I have a checklist protocol that's written out. What are the steps I need to do? And I also try to draw pictures of it, just hopefully to, connect and make it more intuitive. So, this was our daily protocol that the 4 middle school instructors and the undergraduate researcher who partnered with me to study the succession in the sourdough starters across 10 types of flour. This is what they did every day. On step 1 was only the first day. They mixed their flour and water. And then from there on, every day they measured the height when they came in. Then they stirred it up, measured the pH, and then they put 1 milliliter of sourdough starter into it's a snap cap tube. And that we set aside, we put into the freezer, and then we decided at the end which of those samples we were going to send off for DNA sequencing. So that was our in-house graphical protocol.
So did everybody who was following they all sent you the sample for sequencing as well as that's what you used? They sent you the sample. That's how you
Yeah.
did the study. I see. Okay.
Yep.
And then the rest of it, like the backslopping and everything else, that's for their
Yep.
use at home while
That's what they did. Yep.
and of course they were only required to build a starter, but were they also asked to create a bread from their starter?
So I've developed and worked with another citizen science participant to make Dr. McKenney's little loaves for tiny hands recipe. So because we only have 1 tablespoon of discard, I just scaled down, my easiest sourdough that I make every Saturday night to bake on Sunday mornings. And I scaled that way down to a tablespoon of sourdough starter. So that you could, without using tons of flour, you could bake a loaf a day and you could see, oh, we have rock cake, rock cake, rock cake. But by, maybe day 5, it's palatable. Or, at what point does the leavening actually make it through the oven?
Did you get many reports from people that followed through on that aspect of it?
Oh, we got a lot of reports from 4-H in North Carolina. The 4-H agents across the state launched, a little loaves for tiny hands holiday series for sourdough. We got reports that a lot of kids love making a small batch with like just their size of a loaf of bread.
That's so cool.
Yeah.
Did any of them make pancakes? I would think the pancakes would be a great easy entry for kids.
That's also on my list. I like to, share out a list of, these are some of my favorites and they're pretty easy. My 3-year-old can help me. And I use ground flaxseed instead of eggs in my pancakes. So I don't have to worry if she's like sourdough starter,
Yeah, exactly. Yeah. This is well, Mike Nolan asks again, Michael Gonzalez seemed to indicate that multiple feedings per day tend to produce starters that have a high percentage of Lactobacillus San Francisco. I always say San Franciscans. I don't know if I'm saying it right. Did you come to a similar conclusion? Multiple.
So we were controlling the number of feedings. The best way to get to that question, if we wanted to test Gonzalez's theory, right, would be to grow a number of different starters that are, one is fed only once a day, one is fed twice a day, one is fed 5 times a day, right, at all different intervals and frequencies. We held the feeding constant at just 1 refreshing and feeding per day. So we were not able to ask that question.
But that wasn't part of the scope of this project. Yeah.
Right.
Okay. That's good to know. Sugyu Lee asks, would you elaborate more about the influences of different flours to flavors and microbial compositions? Can I say that different flours lead to different cultures or?
Yeah. So here are the graphics for that. This is the heat map on day 14, comparing the bacterial communities across the 10 different flour types that we grew our starters from for the Succession project. Teff is just wild. I mean, to be so highly dominated by Pediococcus and almost nothing else, like Teff really stood out. So I like the this heat map format for really calling out, you can see that all-purpose and turkey wheat and even, emmer and einkorn and rye, they tend to have communities that are split a little bit more evenly, right? You don't have just blocks of red. Whereas Teff is taken completely over by Pediococcus. Amaranth is taken pretty well over by Lactobacillus. Let's see what the captions do with those genera. So I think that heat map helps to highlight the community differences in not only which bacterial genera are present, but also which are more dominant across these different flour types. And then that next figure that I showed is, again, another way of visualizing those different communities. So here, instead of looking at different bacterial types, we're comparing, each point is a community for a sourdough starter grown from a different flour type. And what this shows us is that a rye starter looks like a rye starter, and a sorghum looks like a sorghum, and a teff looks like a teff, but all purpose kind of is encroaching onto einkorn, right? So we can ask about, an amaranth has a lot of variation among those different starters communities. So this graph tells us a little bit more about the relationships and the relative similarity of those different communities to one another. But then as far as the aromatic profiles, that's what we've plotted in bold with dashed line arrows on top. So you can see that, the communities associated with amaranth starters down at the bottom in orange, they tend to produce aromas like meat, like fermented dairy, and like toasted or grain.
Now, Mike asks again, since many bakers started with whole wheat or rye, but then switched to all-purpose or bread, what does that changing flour do to the starter?
Yeah, that's a great question. I can speculate. I can imagine that perhaps using a rye or a whole wheat to start with, we found out from that plating exercise with the high school students I worked with that those whole grain flours do have a lot more and more different types of viable yeast in the flour itself. So it might jumpstart your starter at first, but again, it will set up different interactions between microbial types within that community. But we haven't tested it formally. So yeah, I imagine it could also be kind of a culture shock to go from, wait, I had rye flour for days, and now what is this? What are you giving me now? But no, we haven't in the lab, kind of quantified or studied what that culture shock of the diet change of a flour type might mean.
You managing the number of citizen scientists that you had, it must have been an immense task, and I'm amazed that you were able to gather so much information. But clearly, every time you add another question or factor into it, it's like multiplying it, by another power. So
exactly.
So, yeah. but it should keep you busy for the next part of your life.
Yeah, it's really fun, and the things that I can't scale up to the lab immediately, I test in my kitchen sometimes. That's a really cool idea, Mike. I'm gonna maybe try and grow a starter and then switch its food partway through and see. The microbial ecologist part of me is, because there are many hats, right? My microbial ecologist side is thinking, well, would that kind of push you back in your succession timeline? Because, the community was maybe pushing toward a more stable, mature rye associated community. And then if you switch diet, are you pushing it back to maybe not square one, but square 2 or 3? Does that mean that your mature starter would not reach a stable baking point until day 17 or day 20? I don't know.
Well, this could go on for so long, the research as it comes in. So of course we have to keep having you come back every time we do one of these events for updates. But if people do want to stay in touch with you and get a feed, or is there a newsletter or some kind of a ongoing way that they can be tied in, or is it through Facebook? What's the best way for folks who are interested in staying connected with you and your work?
I would say for folks who are looking for community, the Facebook groups are thriving and vibrant and full of wonderful people who are very transparent and open and sharing. There are no secrets on the Facebook sourdough groups. And there you also have a higher chance if you email me, especially like come August. My semester will be starting, so your email will be fighting for an audience with, my 100 students. That's not to say that anyone is more or less important than anyone else. Just there will be more signal that you're battling. If you are looking for a short-term answer that maybe, a baking another baker if you're looking to bounce ideas off of each other and you're looking for community. Also, Rob and Lauren and Anne and Leah and there are at least 5 of us who are admin on that Facebook group. And so, if you can find it and join it, then you have a 5 times greater chance that one of us will see it.
And what's the name of that group again?
So I think the one is Wild Sourdough for Science.
Because there's a lot of sourdough groups, so you got to get the one that you guys are the admins for. So Wild Sourdough for Science. That's a keywords there. Okay, thanks. By the way, here's a shout out. Thank you for the closed captions from Jean. They're terrific. Thank you for that. You're the first one that's used that. You said it's a feature in. Is it a PowerPoint feature or is it
in Microsoft PowerPoint? You can set it up in slideshow. I just have it ticked to always use sub captions, and then you can select your language as well.
That's incredible. Well, I have to keep that in mind the next time I have a chance to do one of this. We'll end with 2 quick ones. Do time and temperature matter for this study? So,
Bernie, I'll say in the global study, we had no control over anything. We were asking folks, hey, will you tell us about your starter and send us a bit of it? So we were casting the widest net possible, and we had zero control over any of it. So as we were trying to analyze the data, we were thinking, well, maybe part of the reason that we don't get a very high signal of effect of all of the human-centric baking management practices is that we didn't control any of it, right? Which is why in this succession study, when we were studying specifically what is the effect of flour type and the age of the starter from day 0 to day 14, we did grow all the starters in the same lab space. So we tried to have time and temperature not matter in this study.
But when you got it back in the lab, then you had more control.
Right.
Yeah. Finally, Chad Robertson recommends using just 5% starter to inoculate a new starter. So 1 tablespoon of discard would give you quite a bit to work with. Is that 5% seem to be reasonable?
I would say my starter, David Dowie, who is downstairs in our kitchen. I also
David Dowie. Okay.
David Dowie. It's a spoonerism.
Yeah.
I probably use something like only about 5% to inoculate the next when I refresh it. But he's well established, right? David Dowie's a classic, as they say. If I am growing one from scratch, from flour, that's when I'm keeping about 50% of the initial volume to inoculate the next one, right? So I think earlier in life, it may play a more important role to retain more of that developing community. But that's also based off of the King Arthur flour instructions. So again, we tried to, Martha and I had both talked to a number of bakers from across different scales of production and experience levels. And we were also, I was combing the internets to figure out, what are the general practices and kind of the common wisdom around starting a starter from scratch. And so I tend to think if King Arthur has embraced something and is putting it out for the world, it's a pretty safe bet that a lot of people are seeing it and that it works reasonably well.
Yeah. Well, I'll squeeze one more in, even though we've run over time. Sung Yu Lee again said that levain with flour different from that for feeding. And I guess what he's saying is that if the flour used in the levain is different than what's used in the original starter and the mother starters or whatever, a good way to introduce new flavors to bread. The time for levain maturing. Is that enough to make this kind of flavor impact, or is it too short of a span?
That's another great question. Let's see. Yes, it is enough time to have an impact on the overall flavor. I'll say I feed my starter just all-purpose flour for, cheaper maintenance. And on Saturday mornings, I bring, about a tablespoon out into a mixing bowl, and I actually give it then about half a cup of whole wheat flour and about half a cup of bread flour. And then, a little under a cup of water, because I work in volumes most of the time too. So I leverage the levain as a way to transition toward, what flour mixture is Bava doughy going to see, when I'm actually like moving toward a bread dough.
But you all feed David Dowie the same dinner.
The maintenance diet is just all purpose.
That's okay.
So the levain I treat as a transition toward, so that Saturday night when I'm mixing the actual bulk ferment, then it's already seen and experienced and adjusted to in some way the whole wheat and bread flour blend. But I will say, let's see, you have the community in the starter, right? That would be maintained and stabilized with whatever you're feeding your starter regularly on a daily or thrice daily basis. That's the microbial community that's doing all of the work to produce those flavors and aromas. So you can indeed have an impact on flavor, by feeding that stable, mature starter in the levain a different flour mix. But you're just changing it in the same way that if I usually eat salads and lentils, and then tonight I eat, creamy, meat heavy Indian for dinner, like my metabolism is going to be different, right? Thanksgiving dinner or Christmas dinner, right? Every year we all experience this after some meal or special occasion in our lives. That's what you're changing. If you have a short-term flour shift. And again, I haven't sequenced the DNA of these different, bread making stages. So this is speculation. But that seems like too short a term to impact the actual full sourdough community. I would imagine that what you're impacting instead is the microbial metabolic products of digesting that new diet.
Yeah. Well, let's continue this conversation in the after party. I want to invite all of you who can to join us. We're going to log out. Erin and I will log out and log back in, and we'll rejoin in about 3 to 4 minutes. We'll come back in. Most of you can get over there faster than we can because we have to log out and then log in. But and one thing we should definitely discuss when we come back in, depending on how many people we have, is I want to hear the different names that everybody's come up with for their starters. Because David Dowie, we got David Dowie to start the ballroom, but I know some of you have your own starters with some cool names. So we'll share that information when we come back. And I want to thank all of you who have been watching, and those of you who are watching on the archives and are not going to be able to join the conversation later on for watching this. And you know how to get in touch. You can use the dashboard to communicate with anybody who's registered for the symposium. So please feel free to communicate with each other. And we'll see some of you in a few minutes in the after party. Erin, thank you so much for this great presentation. As always, we love having you, and I can't wait to continue to hear how things develop and evolve. And thanks for creating so many young citizen scientists out there for your work with young people as well.
Thanks for having me, and thanks everyone for your questions. It's been a blast.
Okay, we'll see you in the after party. Bye-bye, everyone. Thank you to our team behind the scenes. Our event technical and production partners, Ganoid Communications, including our producer, Gurmit Singh, and his team, Jita Gajaria, Gagandeep Singh, and Jaydev Kashari. Thanks also to Ted Nelson and Lael Fretzel of our creative and marketing team at Gumbo Marketing, and the many folks at Johnson & Wales University who supported me throughout this event. my executive assistant, Sarah Standifer, communications director, Melinda Law, Chancellor Mim Rooney, Charlotte Campus President, Cheryl Richards, and our executive team leaders, deans and faculty, Maureen Dumas, Michael Schrader, Michelle Nicholas, Mark Norman, Brent Steyerwalt, Laurie Heimbach, Jerry Lanuza, Amy Felder, Harry Paymiller, Richard Miskovich, and many, many others. Thank you all.